Hello everyone, welcome back. So we are dealing with evolution, right? In last video lecture, we have seen the theories, the modern synthetic theory or chemogenetic theory. Modern synthetic theory is divided into three groups: chemogeny, biogeny, cogenogeny. In chemogeny, chemical evolution occurs. In biogeny, biomolecular evolution occurs, and then in cogenogeny, the first life form. So the first life form is known as eobion. Go with eobion, okay? Don't go with protobion. Though protobion was first life form, but they were unable to reproduce. So we go with eobion or co-acerbates. So by using certain chemicals and light, they are able to synthesize carbon dioxide and releases oxygen in the atmosphere, and that organism was from eobion. It is developed into cyanobacteria. All right. So that was our theory. Now we will see the experiment performed by two scientists to prove the chemogenetic theory or modern theory of evolution. This experiment was done by Urey and Miller. That is important. Question always come from this experiment. So today we will discuss Urey and Miller experiment on chemogenetic theory, is also known as modern theory of evolution. So these two scientists performed an experiment to prove that the chemicals, whatever the chemicals was evolved. So we need to draw the experimental setup. We're going to see the chemicals that has been used by these two scientists, and we will see the experiment. So for this experiment, Urey and Miller, they use a round bottom flux kind of thing. In NCERT book, the diagram is there, but here I'm going to show it in a simple way. I'll try to make this simple diagram, okay? You can rely on this diagram, as well as if you want more in a specific form, the diagram, you can go through the NCERT book diagram. But here I'll try to show in my own style, or I'll try to make it simple. So he took a round bottle flux. This round bottle flux has a tube connected with it. This tube is connected with another flux. And then the same tube, it goes back into that round bottom flux. We call that structure it as round bottom flux, okay? So that was their experimental setup. So I'm making it a simple diagram. If you want in a more specific form, you can go through the NCERT book. But both are correct. It doesn't matter. Okay. So we're going to name the experimental setup. This round bottom bottom flux, we say bottom. it is connected with an electrode, a positive charge and a negative charge electrode that passes electric current inside the flux. So electrode has been placed, and this structure we call it as a round bottom flask. And this type of structure, which is connected with tube, is known as condenser. In condenser, the water goes in and the water comes out from that side. That one is another flux. Okay, that's it. A very simple diagram which will make you to understand in easiest way. So they took an experimental setup, a round bottom flux which is connected with a tube. The tube has condenser with it. Then this tube has a flux which is connected, and that same tube, it comes back to the same bottle flux. Now for this experiment they used a chemical, methane, ammonia and hydrogen. They use a chemical, methane, ammonia and hydrogen in the ratio of 2:1. This chemical is added inside the flux. The chemical has been placed inside the flux where the two electrodes were connected. They are giving the electric current electrode. So this electrode represents the rays of the sun. It represents the energy. So inside the flux, okay, Urey and Miller, they placed chemical, methane, ammonia and hydrogen. That is very important from entrance point of view. This always asks a ratio also. Methane 2, ammonia 2, hydrogen 1. Okay, and they also added a little amount of water inside the flux, and they put the electrode, two electrodes. That side I'm going to write it down, the important things of the experiment. So they kept the chemicals like methane, ammonia and hydrogen in the form of ratio 2:1. And they performed this experiment for about 7 days. 7 days. So they performed their experiment for about 7 days, and the temperature, they kept it about 400 degrees Celsius inside this bottom flux. Okay, so the temperature they allow inside the bottom flux was about 100 degrees Celsius, and they performed the experiment for about 7 days continuously. So when these electrodes were kept and allowed to pass the electric current, and the temperature they adjusted for 100 degrees Celsius, the reaction occurred inside the bottom flux. A little amount of water also kept, okay, but I'm not writing it here. So little amount of water was also kept. So then the reaction occurred. This reaction, after reaction, they pass whatever the chemical after reaction occurs, they pass the gases through the tube and these gases comes inside this flux. All right. After giving an electric charge, the reaction occurs with the chemical in the presence of light. Electrons represent as a light, okay, energy. So after reaction, this all gases, they comes inside the tube and come at the another flux. Now Urey and Miller, they took out the gases from this flux. They extracted the gases from this flux. Okay. Then they analyze the gases. So they extracted whatever the gases were there in the flux. They extracted all those gases and analyzed that gas. Okay. After analyzing all those gases, they have found out that all those gases are found out to be simple sugars like aldehyde, ketone, amino acid, purine, pyrimidine, etc. Okay, and that the same gas, it goes back inside that round bottom flux. So that is the experiment, very simple. So from this experiment, they have concluded that in the presence of energy, so energy here represented in the form of electron. So in the presence of energy, all those chemical, they reacted, and after reaction, all those chemical has been transformed into a simple sugars like aldehyde, ketone, purine, pyrimidine, and amino acid. Okay, so from that experiment, they have concluded that the theory, that is the chemogenetic theory, explains the same thing. So that's why that theory has been considered as the modern theory of evolution and the accepted theory one. Okay. So what they did, they took a round bottom flux. This round bottom flux has a tube. It goes and connect with the flux, another flux, and from that flux, the tube again goes back into the same round bottom flux. This round bottom flux is connected with two electrodes, and they put a chemicals, ammonia, methane, and hydrogen in the form of the ratio 2:2:1. And this experiment was performed for 7 days, and they have adjusted the temperature inside the round bottom flux for about 100 degrees Celsius. So when electrode was passed inside the flux where there were chemicals, the electrodes, that is the energy, is reacted with those chemicals. Now this chemical pass with the help of the tube and goes inside the flux. So from this flux, they have extracted the chemical and they analyzed the chemical. After analyzing the chemicals, they have found out that from that chemicals, it has been transformed or evolved into a simple sugars like aldehyde, ketone, amino acid, purine, and pyrimidine. Simple. Okay, that is the experiment which is performed by Urey and Miller, and from this experiment they have concluded that the chemogenetic theory is actually a correct explanation which was given by Operin and Helden. What it says, in the presence of light where there was a torrential rain, it reacted with the water molecule and broth formation occurred. So from that, a new simple sugars were evolved. Okay, so it react with water in the presence of light. Here also same. In the light was here in the form of electric charge, and they also added small amount of water molecule here. So in the presence of light with water molecule, all those gases reacted, and after reaction, They have found that they have transformed into a simple sugar. That's how the chemicals start to evolve. So that was the experiment performed by Urey and Miller. Very important. You need to remember is the chemicals. What are the chemicals they have used, and for how much of it a ratio, how many days they have performed experiment, and then temperature. That's it. You can write your own answer, okay? But you have to show that experimental setup. This is just a simple experimental setup, and if you want it more in specific form, the experimental setup, you can go through NCERT book, but this will do still, okay? But if you want more in specific form, those who like drawing, they can go through NCERT book, okay? That's it. So from here they have extracted the gases chemical, they have analyzed it. See, the process of extraction of gases, the analyzing has a process, a long big process. We are not going to talk it here now. Our concern is only to the evolution part, not the technique part, okay? Evolution we need to see. All those technique part we're going to talk about in biotechnology chapter. Bujhi pala? So here to experiment. That's it. All right.